This is the most important photograph in the history of biology. It doesn't look like much. A blurry X shape, some smudges, a pattern of dark spots on photographic film. But this single image revealed the secret of life itself. The code that makes you, you. The instructions that build every living thing on Earth. It took 100 hours of X-ray exposure to capture. It required years of expertise to create. And it answered a question scientists had been asking for decades. What is the shape of the molecule that carries heredity? The woman who created this photograph was one of the most brilliant experimental scientists of her generation. She spent months perfecting the technique. She calculated the dimensions. She knew exactly what she had. She was weeks away from solving the structure herself. And then someone showed the photograph to a competitor. Without her permission, without her knowledge, without ever telling her it happened. She would be dead before the Nobel Prize was awarded. Her name wouldn't be mentioned in any of the acceptance speeches. For decades, history would remember her, if it remembered her at all, as a difficult woman who got in the way of progress. This is the story of how a single image, seen by the wrong person at the wrong time, unlocked the code of life and erased a scientist from history. Part 1. The Players. By 1950, scientists knew something remarkable about a molecule called deoxyribonucleic acid, DNA. They knew it carried genetic information, the instructions for building every living thing, from bacteria to blue whales, were somehow encoded in this long, twisted molecule tucked inside every cell. But they didn't know how. The structure was a complete mystery, and structure matters enormously in biology. Once you understand the shape of DNA, you understand how it works, how it copies itself, how parents pass traits to children, how a single fertilized egg contains all the information needed to build a human being. Crack the structure, and you crack the code of life itself. 3 laboratories were racing to solve it. King's College London had the best X-ray equipment and a new researcher who was an expert at using it. The Cavendish Laboratory at Cambridge had 2 ambitious scientists with an unconventional approach and very little data. And Caltec in California had Linus Pauling, the most famous chemist in the world and the overwhelming favorite to solve the puzzle. Whoever cracked DNA first would change biology forever. They would win the Nobel Prize. They would be remembered for centuries. This is the story of that race, and the woman history tried to forget. To understand what was taken from Rosalind Franklin, you first need to understand who she was. Franklin came from a wealthy London family. Her father was a banker. She could have lived a comfortable life of leisure. Instead, she became obsessed with science. At Cambridge, she studied chemistry, one of a handful of women in a field dominated by men. After graduation, she worked on coal research during World War II, studying the microstructure of carbon. Her work helped Britain develop better gas masks. It was precise, painstaking, and essential. But it was in Paris that Franklin found her true calling. She spent 4 years at a government laboratory there, learning X-ray crystallography. The method is deceptively simple in principle. You fire X-rays at a molecule, and the rays scatter in patterns that reveal the molecule's shape. Like shining a flashlight through a crystal chandelier and studying the pattern of light on the wall. In practice, it's extraordinarily difficult. You have to prepare a sample pure enough to produce a clear pattern. For DNA, this meant extracting fibers so fine they were barely visible, then stretching them and keeping them at exactly the right humidity. Then you expose the sample to X-rays, not for seconds, but for hours, sometimes days. You sit in the dark, monitoring the equipment, adjusting the angle, waiting. And when it's done, the resulting image isn't a photograph of the molecule. It's an abstract pattern of dots and smudges, a kind of shadow cast by invisible atoms. To interpret it, you need sophisticated mathematics to work backward from the pattern to the shape. Most scientists found crystallography tedious. Franklin found it beautiful. She was exceptional at it. In Paris, she made important discoveries about the structure of coal and carbon. She published papers that established her reputation. She developed techniques that were years ahead of her time. Her notebooks were legendary, so meticulous they could have been published as is. Every measurement recorded, every observation documented with obsessive precision. She was 30 years old, at the peak of her abilities, and one of the world's leading experts in using X-rays to reveal the hidden structures of molecules. King's College London hired her to tackle one of the biggest puzzles in science, the structure of DNA. She didn't know there was a problem waiting for her. The problem was a man named Maurice Wilkins. Wilkins had been working on DNA before Franklin arrived. When he heard the laboratory was hiring a new researcher, he assumed she would be his assistant. She wasn't. She was hired as his equal. Wilkins never accepted it. From the moment Franklin arrived, the relationship was toxic. He resented her independence. She resented his condescension. They were assigned to work on the same problem in the same laboratory, and they could barely stand to be in the same room. Within months, they had stopped speaking entirely. They communicated through formal memos. They divided up the DNA samples. She took the wet form, he took the dry form. They worked in parallel, in silence, in mutual contempt. But the problem went deeper than Wilkins. King's College in 1951 was not a welcoming place for women scientists. The senior common room, where researchers gathered for lunch and conversation, was men only. Franklin was excluded from the informal discussions where ideas were shared and collaborations formed. In science, isolation has a cost. You need collaborators. You need people to bounce ideas off, to challenge your assumptions, to see what you might be missing. You need to be in the room where it happens. Franklin had her data, but she was alone. 60 miles away in Cambridge, a very different approach was taking shape. While Franklin spent months preparing samples and capturing photographs, two men at the Cavendish Laboratory had decided that experiments were mostly a waste of time. They weren't going to do the hard work of collecting data. They were going to guess. James Watson, American, ambitious, impatient, absolutely convinced he was destined for greatness. He'd been a child prodigy, enrolled at the University of Chicago at 15, earned his PhD by 22. He was brash, competitive, and utterly uninterested in the slow, methodical work of experimental science. He found lab work boring. He wanted to think big thoughts and make big discoveries. Watson wanted glory, and he wanted it fast. Francis Crick, British, loud, brilliant, impossible to ignore. When Crick walked into a room, everyone knew it. He had an enormous laugh and an even bigger ego. He would interrupt seminars to correct the speaker. He would wander into colleagues' offices and tell them their research was wrong. He was, by his own admission, obnoxious. And despite being 12 years older than Watson, he still hadn't finished his PhD. The war had interrupted his career. He'd worked on naval mines, then drifted between projects, never quite settling down. He had a reputation for being brilliant but unfocused. But he was genuinely, spectacularly smart. He saw patterns that others missed. He made intuitive leaps that turned out to be correct. When Watson and Crick met, something clicked. They were both outsiders. They were both convinced that DNA was the most important problem in biology, and they were both willing to take shortcuts that more careful scientists wouldn't. Their method wasn't experimentation, it was model building. They would take what was known about DNA's chemistry and try to physically construct a shape that fit the data. Metal rods for bonds, cardboard cutouts for bases, scientific tinkering. And it had already failed once. In late 1951, Watson and Crick built their first model of DNA. They invited researchers from King's College to come see their triumph. It was a disaster. Franklin took one look and tore it apart. The chemistry was wrong. The water content was wrong. The dimensions didn't match the X-ray data. Complete nonsense. She was right. The model was so bad that the head of the Cavendish Laboratory ordered Watson and Crick to stop working on DNA entirely. For Watson and Crick, it was humiliating. They'd been caught guessing, and they'd guessed wrong. But they didn't stop thinking about DNA. The problem was too important. The prize was too big. They just needed better data. Part 2. The Race. By early 1952, the race for DNA had settled into a strange equilibrium. Franklin was methodically collecting the best X-ray data in the world, but she wasn't sharing it. Watson and Crick were forbidden from working on DNA, but they couldn't stop thinking about it. And Linus Pauling was an ocean away, working on his own model. Everyone assumed Pauling would win. You have to understand who Linus Pauling was to understand how frightened everyone was of him. He was the greatest chemist of his generation, possibly the greatest of the 20th century. He had already revolutionized how scientists thought about chemical bonds. He had written the textbook on the subject. He had discovered the alpha helix, the fundamental structure of proteins, in a breakthrough that would win him the Nobel Prize. His track record was terrifying. When Pauling focused on a problem, he didn't just solve it, he demolished it. And now he was interested in DNA. Watson and Crick followed Pauling's work obsessively. Every rumor from California sent them into a panic. Every hint that Pauling was making progress felt like a countdown clock. In late 1952, the news they dreaded finally arrived. Pauling had written a paper. He had solved the structure of DNA. The race was over. They had lost. And then they read the paper. Pauling's model was a triple helix. 3 strands wound around each other with the phosphate backbone running through the center. It was elegant. It was clever. It was also completely, catastrophically wrong. Phosphate groups carry a negative electrical charge. Pack 3 of them into the center of a molecule and they would repel each other violently. The structure would tear itself apart. The greatest chemist in the world had forgotten freshman chemistry. How could Pauling have made such a basic error? He was rushing. He wanted to beat the British teams. And crucially, he was working with bad data. Old, blurry X-ray images from 20 years earlier. He didn't have anything like the crisp photographs Franklin was producing. Watson and Crick suddenly had a reprieve. Pauling would realize his mistake within weeks. But for now, the race was back on. While Pauling was publishing his flawed model, Rosalind Franklin was doing what she did best. For months, she had been refining her technique, adjusting the humidity in her camera, perfecting the preparation of DNA samples. In May of 1952, she and her graduate student Raymond Gosling produced something remarkable. They called it Photograph 51. The image was unlike anything anyone had seen before. A stark X shape dominated the frame. The pattern was crisp and clear. The spacing between the bands was precise and measurable. To a trained eye, the photograph screamed helix. It was the clearest X-ray image of DNA ever captured. It would become the most important photograph in the history of biology. Franklin knew what she had. In her notebooks, she wrote that the structure was probably helical. She calculated the diameter. She figured out how many nucleotides fit in each turn. She determined that the phosphate backbone must be on the outside of the molecule. She was so close. But Franklin was a careful scientist. She wanted more evidence. She wanted to be certain. She decided to wait. That decision would cost her everything. Part 3. The Betrayal. January 1953. James Watson visited King's College London. Officially, he was there for a seminar. Unofficially, he was fishing for information. Watson and Franklin did not get along. She was precise, reserved, rigorous. He was brash, impatient, dismissive. Their conversation that day quickly turned into an argument. Watson left her laboratory frustrated and empty-handed. But then Maurice Wilkins pulled him aside. Wilkins was fed up with Franklin, fed up with the tension, and he wanted to show Watson something. He took Watson to another room. He opened a drawer, and he pulled out photograph 51, Franklin's photograph, her work, her data, the product of years of expertise and months of careful preparation. He showed it to her competitor, without her permission. James Watson looked at the photograph for less than a minute. It was enough. Years later, he would describe the moment. The instant I saw the picture, my mouth fell open and my pulse began to race. The X pattern was unmistakable. Everything he needed to know was right there, captured in a single image. Watson rushed back to Cambridge. But the photograph wasn't the only thing Franklin's competitors would see. A few weeks later, Max Perutz, a senior Cambridge scientist, shared something else. King's College had submitted a progress report to the Medical Research Council. The report contained Franklin's unpublished data, her calculations, her analysis. Perutz gave the report to Crick. Franklin had spent years developing the techniques to capture photo 51. She had spent months analysing the data. Now Watson and Crick had all of it. Her images, her numbers, her ideas. They had everything they needed. Part 4. The Discovery. February 1953. The Cavendish Laboratory, Cambridge. Watson and Crick worked feverishly. They knew DNA was a helix. They knew the phosphate backbone was on the outside, but something wasn't clicking. The problem was the bases. DNA contains 4 chemical bases: adenine, thymine, guanine, and cytosine, A, T, G, and C. These bases had to be on the inside of the helix, connecting the 2 strands. But how? Watson made cardboard cutouts of the bases and moved them around on his desk like puzzle pieces. Nothing worked. The breakthrough came on a Saturday morning, February 28, 1953. Watson was playing with his cutouts again. For weeks, he had assumed that identical bases paired together, A with A, T with T. It seemed logical, but it didn't work. The pairs came out different sizes. That morning, almost by accident, he tried something different. What if different bases paired together? Suddenly, he saw it. Adenine paired with thymine, guanine paired with cytosine, A with T, G with C. And here was the beautiful part. Each pair was exactly the same size. A large base always paired with a small base. The rungs of the ladder were perfectly uniform. 2 strands twisting around each other in a double helix. Base pairs connecting them in the middle. A shape that was elegant, symmetrical, and self-explanatory. Because if you unzipped the 2 strands, each half contained all the information needed to rebuild the other. A knows to pair with T, G knows to pair with C. The molecule could copy itself perfectly, over and over, forever. This was how heredity worked. This was how parents passed traits to children. This was how a single cell contained all the instructions for building a human being. It was beautiful, it was simple, and it changed everything. On that Saturday morning, Watson and Crick knew they had solved the most important problem in biology. They had discovered the secret of life. That afternoon, Francis Crick walked into the Eagle pub in Cambridge. He announced to everyone present, "We have found the secret of life." No one believed him. Crick was known for his enthusiasms, his grand pronouncements, but this time he was right. Part 5. The Aftermath. On April 25, 1953, 3 papers appeared together in the journal Nature. The first, by Watson and Crick, described the double helix model. 900 words. It would become one of the most important scientific papers ever published. Near the end, a single sentence acknowledged they had been stimulated by unpublished results from King's College. One sentence. That was all. Franklin's paper was published alongside, but framed as supporting evidence, not as the foundation that made the discovery possible. She was reduced to a footnote in her own story. A few weeks later, Franklin visited Cambridge to see the model. She examined it carefully. She checked it against her data, and she recognized immediately that it had to be right. What happened next was surprising. Franklin didn't make accusations. She didn't complain about how her data had been used. She simply accepted the result and moved on. Over the following years, she and Francis Crick became genuine friends. She vacationed with him and his wife Odile in Spain. After King's College, Franklin moved to Birkbeck College, where she built a new research group studying viruses. Her team produced groundbreaking work on the tobacco mosaic virus, research that laid the foundation for modern virology. She was doing the best work of her career. She was also dying. In 1956, Rosalind Franklin was diagnosed with ovarian cancer. She was 36 years old. The cause was almost certainly the X-rays. Years of exposure to radiation, the same radiation that had helped her capture photo 51, had damaged her cells and triggered the cancer that would kill her. In those days, researchers didn't fully understand the dangers. She kept working through the illness, through surgeries, through experimental chemotherapy that left her exhausted and sick, through periods of remission that gave her hope, and relapses that took it away. In her final 2 years, she published 17 papers. 17. While fighting cancer, while knowing she was probably dying. She traveled to conferences, she mentored students. She built intricate models of viruses for an international science exhibition. Her friends begged her to rest. She refused. There was too much left to do. On April 16, 1958, Rosalind Franklin died. She was 37 years old. She never received proper credit for her contribution to the discovery of DNA. She never knew how history would eventually judge the scientists who used her work. She never had the chance to tell her side of the story. In 1962, the Nobel Prize in Physiology or Medicine was awarded to James Watson, Francis Crick, and Maurice Wilkins. The Nobel Prize cannot be awarded posthumously. Rosalind Franklin's name was not mentioned in any of the acceptance speeches. 6 years later, James Watson published his memoir of the discovery. He called it The Double Helix. It became an immediate bestseller. It was praised for its honest, gossipy account of how science really works. The competition, the personalities, the race for glory. It also shaped how a generation understood the discovery of DNA. Watson described Franklin as Rosie, a nickname she never used and would have hated. He described her as dowdy and difficult. He suggested she didn't understand her own data. He portrayed her as an obstacle to be overcome, not a scientist whose work made the discovery possible. The book was devastating. Watson would eventually walk back some of his characterizations. In an epilogue added to later editions, he admitted that Franklin's work had been essential. He acknowledged he had been unfair. But the damage was done. The narrative was set, and Franklin wasn't alive to correct it. Part 6. The Legacy. It took decades for the truth to emerge. Historians re-examined the evidence. Biographers dug into Franklin's notebooks. Scientists who had known her spoke up about what she was really like. Brilliant, funny, warm to her friends, passionate about her work. The real Rosalind Franklin emerged. A woman who loved hiking in the mountains and traveling to new countries, who threw dinner parties and kept up lively correspondence with friends around the world, who was fiercely loyal and deeply kind to the people she cared about. A scientist who was not just competent, but exceptional. Slowly, the story changed. Today, Rosalind Franklin is recognized as essential to the discovery of DNA. Her photograph, Photo 51, is famous. Her name is on buildings, awards, and research institutes around the world. The European Space Agency has named a Mars rover in her honor. It's called Rosalind Franklin. When it launches in 2028, it will travel to the Red Planet to search for signs of life, searching for other forms of DNA. The woman who captured life's secret will become part of the search for life beyond Earth. Her name is finally where it belongs, at the center of one of the most important discoveries in human history. Think about everything that has come from this discovery. Every breakthrough in genetics traces back to this moment. Gene therapy that cures diseases once thought untreatable. DNA forensics that has freed innocent people from prison and brought killers to justice. Genetic engineering that has transformed agriculture and medicine. The Human Genome Project, CRISPR, the ability to read and edit the code of life itself. All of it builds on the foundation laid in 1953. All of it descends from the discovery of the double helix, the secret of life, captured in a photograph by a scientist who deserved so much more than history gave her. Now you know how we discovered DNA, and now you know what it cost. If you want to see how we discovered other secrets of the universe, subscribe.